Hello, it is a pleasure for us to be participating in this symposium. I am Andrea Montihius, and together with Veronica Tribilin, my colleague, we are going to share with you the results of our project entitled Translational Bnct Studies in the Hamster Cheek Pouch Oral Cancer Model and in an ectopic model of colon cancer in BD9 rats, employing oligofucoidan to reduce toxicity and or improve Bnct therapeutic efficacy. This work was performed in collaboration with HiQ Marine Biotech from Taiwan and the National Atomic Energy Commission from Argentina. As I mentioned before, we are from Argentina. Here you have some photographs to know a little bit about our country. And here we are. This is our group. And we work, as I said before, at the National Atomic Energy Commission in Buenos Aires City. And we also collaborate with other groups from Argentina and from abroad. Since 2001, our group has devoted its efforts to study new strategies to optimize Bnct and reduce severe induced effects. So, what is Bnct? Bnct stands for boron neutron capture therapy, which is a tumor-selective particle radiotherapy. It is based on the administration of boron carriers incorporated preferentially by tumor cells, followed by irradiation with a thermal or epithermal neutron beam from nuclear reactors and accelerators. The capture reaction between a boron-10 nucleus and a thermal neutron produces high-energy alpha and lithium particles. These particles have high linear energy transfer, high relative biological efficacy, and a short path length of approximately the diameter of one cell. In this sense, damage is circumscribed to tumor cells where the boron-10 atoms are preferentially localized, sparing normal surrounding tissue. Bnct involves biochemical rather than geometrical targeting, being potentially useful to treat all tumor cells populations, including undetectable micrometastasis and foci of malignant transformation in field cancerized tissue. Bnct is a non-mutilating organ-preserving therapy that exerts a significant tumor control with good quality of life, as it is usually administered in a single session or at most two, in cases in which no other therapy could be useful. Several pathologies have been treated with Bnct, being head and neck tumors and glioblastoma multiforme the most studied targets. We are part of an international society of neutron capture therapy. Here you have the info related to the web page of our society. You can subscribe to our newsletter to know more about Bnct and be updated. Head and neck cancer has high incidence, prevalence, and mortality. One of the most frequent sites is the oral cavity. Our group has extensively studied Bnct in the hamster cheek pouch oral cancer model. It is widely accepted for the study of oral cancer and mucositis, a common toxic effect induced by cancer therapies. It consists of the topical application of the MBA, a chemical carcinogen, in the hamster cheek pouch during 6 to 12 weeks, depending on the model studied. This chemical cancerization protocol closely mimics events involved in the development of precancer and malignant human oral lesions, provides a tumor model surrounded by precancerous tissue, which gives rise to the formation of additional tumors, and allows for the study of mucositis as occurs in field cancerous oral mucosa in head and neck cancer patients. In previous studies, in the oral cancer and precancer model induced by the 8-week cancerization protocol, we demonstrated the therapeutic effect of Bnct mediated by the boron compound boron phenylalanine, BPA, and we optimized Bnct when we combine Bnct with oligofucoidan. It is known that oligofucoidan has anti-cancer and immunomodulatory effects. Oligofucoidan enhanced the therapeutic effect of Bnct on tumors to 94% from 67% at 1 month after treatment. This enhanced effect was also seen at 3 months after Bnct. The percentage of tumors with overall responses for the Bnct plus oligofucoidan group was 81% versus a 42% observed in the BPA-Bnct only group. It is also known that oligofucoidan is considered a prebiotic agent. Previous studies showed that oligofucoidan increased beneficial microbiota in the gut. This year, the microbiome, which consists of the bacteria, fungi, viruses that are linked to organs and tissues has been included as a new hallmark of cancer. It may contribute to carcinogenesis as well as shaping cancer immunosurveillance and response to therapy. It is interesting that the hamster cheek pouch is also modeled to study oral microbiota, as previous studies show that the abundance of bacteria in the hamster cheek pouch is similar to that found in humans at phylum level. In this sense, our aim was to explain the potential benefits of combining BPA-Bnct with oligofucoidan. Particularly, we evaluated the potential differences in absolute boron uptake and microdistribution induced by oligofucoidan and the influence of oligofucoidan Bnct and Bnct plus oligofucoidan on hamster cheek pouch oral microbiota. We wondered if oligofucoidan is capable of increasing beneficial oral bacteria and thus enhance Bnct therapeutic effect. Biodistribution and microdistribution studies were performed in 8-week cancerized hamsters. We evaluated two groups, BPA and BPA plus oligofucoidan. Oligofucoidan was topically administered in the hamster cheek pouch the day before and coupled with BPA administration. 3 hours after BPA injection, samples of blood, tumor, precancerous tissue, normal pouch tissue, kidney, and liver as references were taken. They were digested and then measured by ICP-OS to obtain their corresponding absolute boron concentrations for each tissue and organ. For qualitative and quantitative neutron autoradiography studies, tumor, precancerous, and normal tissue from the pouch were fixed with liquid nitrogen, sectioned and mounted on polycarbonate nuclear detectors to be irradiated. Then they were chemically etched and then they were seen in microscope. For microbiota studies, samples were taken with a sterile swab. DNA is extracted from the sterile swab and bacterial communities were characterized by metagenome amplicon sequencing. As to our results, we observed that oligofucoidan administration exhibited a slight tendency to increase absolute boron concentration in the tumor and precancerous tissue. Although this difference it is not significant due to a large spread of the boron absolute concentration values. As to the microdistribution studies, we observed that the autoradiographic images present similar contrast when analyzing diverse histological areas. Those are parenchyma and stromal areas. However, we observed darker images with high boron uptake in the BPA plus oligofucoidan group versus BPA group. Quantitative studies evaluating the ratio, the optical density ratio between BPA plus oligofucoidan and BPA alone were consistent with this tendency obtained by ICP-OS. As to the microbiota studies, we first characterized microbiota from normal tissue and tumor and precancerous tissue after 8 weeks and 12 weeks cancerization. Taxonomy-based analysis at phyla level showed that microbiota composition and proportion changed during cancerization. Particular for the phylum Firmicutes, there was a variation of classes and families throughout the cancerization process. We observed a significant increase in the Bacteroidea class in the precancerous tissue and tumor surface, and only at 12 weeks cancerization in tumor and precancerous tissue we reported the appearance of Fusobacteria class. Previous studies in vitro and in vivo showed a tight relationship between this class, Fusobacteria class, and the development of head and neck cancer. We concluded that oligofucoidan could be enhancing Bnct anti-cancer effect by combining the increase in boron concentration in tumor and its cancer and microbiota modulatory properties. As we were able to characterize oral microbiota in tumors and precancerous tissue of the hamster cheek pouch, ongoing studies are aimed at evaluating the effect of Bnct, oligofucoidan, and Bnct plus oligofucoidan on the hamster cheek pouch oral microbiota. Strategies that could select anti-tumor microbiota will surely help to enhance Bnct. The selection of beneficial microbiota have an important role in oral health, but also systemic patient health. And now we continue with my colleague Veronica Tribilin.
The aim of this study were evaluate the therapeutic efficacy and radiotoxicity of combined Bnct alone or in combination with oligofucoidan or glutamine compared to the standard BPA-Bnct protocol, and also evaluate the abscopal effect, the systemic immune response expressed in the spleen for the combined Bnct group alone or in combination with oligofucoidan and or glutamine. Schematic representation of experimental protocol. We first inoculate subcutaneously in the right hind flanks benign rat syngeneic colon cancer cells. 4 weeks later, born compound was administered to tumor-bearing rats intravenously. 3 hours post-administration of born compound, rats were irradiated at the R3 nuclear reactor, localized in Argentina. 2 weeks post-Bnct, another inoculation of colon cancer cells is performed in non-irradiated contralateral leg, which will allow us to monitor the abscopal effects. At the end of the experiment, the animals were sacrificed in the spleen with removed in sterility for the cytotoxicity and cytometry study of immune cells. The experimental group of this study were combined Bnct mediated by the combined administration of BPA and CB10 and irradiate at R3 nuclear reactor. Combined Bnct plus oligofucoidan is the same as combined Bnct plus oligofucoidan 5 times per week for 7 weeks. And combined Bnct plus glutamine is the same as combined Bnct plus glutamine once a week for 7 weeks. And BPA Bnct is administered only BPA and irradiate an R3 nuclear reactor. And shams group tumor-bearing with the same control with same manipulation, non-treated. During the 7 weeks of treated, glutamine was placed as a West compress once a week, and oligofucoidan was administered orally and topically 5 times a week. Local effect. At the 7-week post-treatment, the ratio of tumor volume between post-Bnct and pre-Bnct was significantly lower for all the groups treated with Bnct versus sham. Using the endpoint, incidence of tumors that underwent a reduction to less than 50% of the initial tumor volume to further assessment therapeutic response, results were 62% for combined Bnct alone, 82% for combined Bnct plus glutamine, 33% for combined Bnct plus oligofucoidan, and only 30% for BPA Bnct. Combined Bnct has a higher percentage of animals than BPA Bnct that undergo a reduction in tumor volume by half or more. This effect is increased when oligofucoidan and glutamine are applied. Lymph nodes regional effects. The regional effect will combine the abscopal effect that results from local tumor treatment of the local effect and radiation in the exposed area. We observe a statistical significant inhibition in the development of lymph nodes metastasis in combined Bnct versus sham, combined Bnct plus oligofucoidan versus sham, and combined Bnct versus BPA Bnct. Radiotoxicity effects. The incidence of severe dermatitis at 2 weeks when the peak occurred was 100% for BPA Bnct, while for combined Bnct plus oligofucoidan, and combined Bnct plus glutamine, it was below 70%. This difference became statistical significance. Although bone concentration were higher in skin for combined Bnct compared to BPA Bnct. Dermatitis was lower. When we look at the recovery of the third week, we see that the severe dermatitis is present in half of the animal which combine Bnct and only in a quarter or less of the animal treated with combined Bnct plus oligofucoidan and combined Bnct plus glutamine. This will indicate an improvement in recovery in the group treated with oligofucoidan and glutamine as adjunct. For the evaluation of the abscopal effect, we identified the animals that presented a tumor volume less than 50 mm cubic in the left leg as an arbitrary cutoff point for each experimental group and calculate the corresponding percentage. A significant difference could be seen in the contralateral leg between combined Bnct versus sham, and a marginally significant difference can be seen between combined Bnct plus oligofucoidan group versus sham. Systemic immune response, the percentage of animals with cytotoxicity level above the mean for the shams group revealed that the immune response was enhanced for combined Bnct versus BPA Bnct. In addition, oligofucoidan seemed to improve the immune response further. Regarding the evaluation of the immune cells population in spleen, a systemic level statistical significant increase in CDH positive immune cells for combined Bnct plus glutamine versus sham, and in natural killer for combined Bnct versus sham were observed. Based on this result, we can infer that combined Bnct improves therapeutic efficacy and reduces radiotoxicity compared to standard BPA Bnct, activating a systemic immune response. Oligofucoidan and glutamine may contribute to enhance these effects. Thank you very much.